Hi everyone, this is lecture 12 on the role of the peripheral nervous system in sensation. We spent most of the time determining what parts of the brain uh, and what parts of the nervous system are involved in motor and sensory effects, but now we're going to get a little more detail with the role of the receptors. In this lecture, we're going to talk about the properties of sensory receptors. Although primarily on the general receptors, we'll mention a little bit about the specific receptors, but these will be mostly covered in later lectures. We'll talk about how we classify receptors, such as exteroreceptors, interoreceptors, and proprioceptors. We'll look at the function and location of receptors for touch, pressure, temperature, pain, and proprioception. And some of these terms we'll cover in terms of adaptation, generated potential, receptive field, sensation, and perception. Okay. So in order for us to discuss about the peripheral nervous system and sensation, we need to discuss about sensory receptors and their neurons associated with them. Okay. So how does the receptors are receptive to different stimuli? Well, first thing we need are the sensory receptors of the peripheral nervous system, and these detect the stimulus. The stimulus can be pressure, touch, heat, as well as light, for example. They need to be transmitted by the peripheral nervous system by the sensory neurons to the CNS. This is what we've been discussing up to now. Finally, the central nervous system neurons has to receive and interpret this information where it's integrated, and has to decide how it's going to respond to it. Okay. We have generally two classes of senses. We have the general senses, which we'll cover today, and we also have special senses. The general senses are specialized receptor endings that detect touch, temperature, or pain, among other things. The special senses are those that detect light, chemicals, or sounds in the environment. Now, the actual sensory receptors are regions of nerve endings that convert the stimulus into electrical signals. This process of the nerve endings converting the stimulus into electrical signals is called sensory transduction. And we'll talk a little about what occurs in that process as well. Okay, a little more about the sensory receptors. The sensory receptors may be encapsulated, meaning they are surrounded by these specialized supportive cells around the nerve endings, or they can be very simple and they're just free nerve endings. Some of these you discussed in SCB 203 when you were talking about the integumentary system. Now, remember our terminology of sensory transduction. This is a process of converting stimuli into electrical signals. So whether it's touch, pressure, temperature, light, converting this into electrical signals in terms of action potentials is called sensory transduction. Transduction is the conversion of one form of communication to another. Okay, what happens during sensory transduction? Well, in most cases, prior to the stimulus, the ion channels of the somatic sensory neuron are closed. Okay. This is accepted for most types of receptors. Later on, when we get to the special receptors, we'll find that this may not be completely true. But for now, prior to the stimulus, the ion channels of the sensory neuron are closed. When the stimulus is applied, mechanically gated sodium channels open, producing a receptor potential. Now, this receptor potential is a kind of a new term, but you're going to find that it's very similar to the graded local potentials we talked about before in function. Now, if the sodium channel is open and there's enough sodium that enters to reach threshold, then an action potential is produced. So in other words, just like gradient local potentials in synapses, sodium channels produce a receptor potential, which may or may not produce an action potential. It all depends if enough sodium enters to reach threshold. Okay, in this figure, we show the beginning of a sensory transduction pathway. In this case, we're going to be looking at a mechanoreceptor in which this probe is pressing down on a person's finger. If you notice, it hasn't touched it yet. And so at this moment, the somatic sensory neuron, which has free nerve endings here, and the sodium channels, or these mechanically gated channels, are not open, they're closed. Now in this figure, the probe is now touching the fingertip. This is going to be stimulating the mechanically gated sodium channels, and due to the pressure, these channels open. And now sodium is entering the channel. Now, if enough sodium enters the membrane and it depolarizes to reach the threshold, then an action potential will be formed. So in a way, it's very similar to what we showed before, the synapse. However, we're not looking at neurotransmitters here, we're looking at ions. Now, the different ways of classifying sensory neurons. One way of classifying neurons is whether their location in the body. And these are the extoreceptors and interoceptors. And based on these prefixes here, you can get an idea of what they mean. Extoreceptors are closely to the body's surface and detect stimuli external to the body. Interoceptors are within the body and detect stimuli originating inside the body. So this is a very general way of classifying receptors, whether they're inside the body or close to the surface. You can also classify receptors by the stimuli they detect. So for example, our examples so far were mechanoreceptors. And we know that mechanoreceptors are stimulated by physical deformation, pressure, touch, deep pressure, vibrations, stretch. The other ones are fairly obvious by their names. Thermoreceptors, they stimulate the cold and hot temperatures. Okay, there are separate receptors for heat and cold. Chemoreceptors detect chemical stimuli. You learned in SCB 203 that there are certain receptors that respond to oxygen, for example, and carbon dioxide. Photoreceptors are going to respond to light. So we're going to talk more about this when we cover the eye in later lectures. The one you may not be familiar with are nociceptors, and these are pain receptors. Okay. Let's look at the different types of mechanoreceptors. And many of these you learned already in SCB 203. For example, Merkel cell fibers or Merkel cell discs, Meissner's or tactile corpuscles, Ruffini endings, and piscine or lamellated corpuscles. So many of these are named after people, and that's why they're capitalized. Also, hair follicle receptors and proprioceptors. Let's take a look at Merkel cell fibers. These nerve endings are surrounded by a capsule of Merkel cells. And if you remember, they're located in the epidermal ridges and are dominant in the fingertips. And another way of characterizing receptors are how fast they adapt. So those that are slowly adapting will continue to receive the stimulus and it'll be detected. Those that are quickly adapting, after a while, they will stop responding to the same stimulus. So for example, if you've worn a hat before, At first, the hat is stimulating the receptors, and you know you're wearing the hat. But after a while, the receptors will adapt, and you don't realize the hat is on your head anymore. These slowly adapting receptors, called Merkel cell fibers, are important for detecting form and texture. So in other words, we can tell things are flat, round, rough, or smooth, and these are important because they're on our fingertips. So in our, as a review, we have a picture here of the skin showing the Merkel cell fibers that are surrounding the free nerve ending. And remember, therefore, discriminative touch with spatial resolution. So there goes for fine resolution of discrimination of shapes and objects. Meisters or tactile corpuscles are found in the dermal papilla. And unlike the Meisters, unlike the previous receptors, these are rapidly adapting receptors. Okay, and they're more numerous than Merkel cell fibers. They're also used for discriminative touch stimuli, but they're not as fine. In other words, the resolution of objects that stimulated will not be as precise as in Merkel cell fibers. So let's look at the tactile corpuscles, and you can see that they're in the dermal papilla here. They're for discriminative touch with less spatial resolution. They're not quite as fine as the Merkel cell fibers. Now, if we go deeper, we have Ruffini endings, which are also known as bulbous corpuscles. And these are located in the dermis, hypodermis, and even in ligaments. And these are slowly adapting receptors that respond to stretch and movement. And unlike the other previous two, they do not transmit discriminative touch stimuli. So they're more of a general pressure receptor. So here are the spindle-shaped Ruffini endings here, deeper into the dermis. And these are for stretch and movement. Finally, we have the lamellated or Pacinian corpuscles. And these are deep within the dermis. And these are kind of unique because they have like a layered onion-like appearance that look like a little sucker. And these are rapidly adapting deep pressure receptors. And this is what you really would want in very deep pressure receptors. You don't want them responding all the time because that would be kind of annoying. So they are rapidly adapting. In this figure here, we see the lamellated corpuscles with the many layers of the plasma membrane surrounding it. And they're important for vibrations and deep pressure. So many of these mechanoreceptors you learned in SCB203. Another receptor you might have learned in SCB203 are the hair follicle receptors. These are free nerve endings that wrap around the hair follicles in the dermis or hypodermis. And these will respond to anything that causes the hairs to bend. So I could not find a picture of it in your textbook, but all I can find is the picture of the hair root. And so the free nerve endings would be wrapping around the hair root here. And so that they will be stimulated by anything that moves the hair. So when a fly lands on your arm and it moves the hairs, you know your fly has landed on you and you shoo it away. Now, the final one that you may have heard about in SCB203 are proprioceptors, because they're located in the musculoskeletal system. And they're important for maintaining the position or the joint or part of the body and detecting movement of the body in space. Because it's important to monitor all ongoing movements. So your central nervous system knows where each body part is in the space. Okay. One way of demonstrating this is the balance control we have when we're standing on one leg. Okay. For a few minutes, most people can stand on one leg with no problem. But if you're asked to stand on one leg for, let's say, a half hour, after a while, you'll get this exaggerated response of your proprioceptors trying to maintain the body in space, and you'll start, jerking back and forth to maintain balance. And this is an excellent way to show proprioception, as your body tries to maintain balance. Thermoreceptors, we mentioned there are hot and cold receptors. They're separate protein receptors. Okay, as you can expect, cold receptors are at lower temperatures from 10 to 40 degrees centigrade. Hot temperatures are warmer at 32 to 48 degrees. Okay, and notice that there's an overlap in the temperatures, and you can do experiments to show that there are two separate receptors, because one will feel cold and one will feel hot. The cold receptors are in the superficial dermis, and the hot receptors are in the deeper dermis. And they both consist of knobs at ends of free nerve endings. Now, what happens when the temperature goes lower than 10 degrees or higher than 48 degrees? Well, a different type of receptor is involved in these cases. And these are the nociceptors, which are the pain receptors. Because at very cold temperatures and very hot temperatures, those are more painful stimuli. Okay. Let's take a look at the structure and receptive fields of sensory neurons. Okay. Now, remember, sensory neurons are what are called pseudo-unipolar neurons with one cell body and two axons. And if we look at the structure of them, as we've shown before, it gives a good explanation of why we need pseudo-unipolar neurons in this particular case. The cell body, as you remember, is in the posterior root ganglion. So here are the free nerve endings with the receptors. We have a long axon, and we know that the cell body of these sensory neurons are in the posterior root ganglion. And then we have another axon that extends out to synapse in the posterior root gray matter of the spinal cord. So these sensory neurons are pseudo-unipolar neurons. And so the receptors will detect the stimuli, and if it's sufficient to generate an action potential, the action potential will be propagated along the axon to the cell body, and then along the axon to synapse within the central nervous system. And then here it can ascend up to the brain. Now, the structure of sensory neurons, although not illustrated in detail in the previous figure, the axons are larger in diameter and myelinated. And so if you recall from our previous lecture that the larger diameter and myelinated axons conduct faster than those that are smaller in diameter and less myelinated. So these sensory neurons conduct fairly quickly. Another term that we use for these external receptors, such as this in the skin, are the receptive fields. Okay, and this is the area that's served by that neuron. So, for example, mechanoreceptors in the fingertips have many sensory neurons, each with a small receptive field. Generally, there are more receptors, more densely populated or densely spread out with a small receptive field. Whereas in the forearm, is innervated by a smaller number of neurons, each with a larger receptive field. Now, the difference between these two is that having a smaller receptive field, you have much more higher resolution of the stimulus that's activating these receptors. Similarly, in the forearm, with fewer receptors, each with a larger receptive field, you don't have quite that fine resolution as you do in, let's say, your fingertips. So in this illustration, we see two areas of skin. One is from the fingertip and one is from the forearm, and indicating their receptive fields. In the fingertips, we see more highly densely spread out receptors, each with smaller receptive fields. In the forearm, we have fewer receptors, but each with a larger receptive field. Now the question, how does this translate into reception? So in this next figure, we have what's called the two-point discrimination. And it's a way of measuring the size of the receptive fields. And the two-point discrimination is the minimum distance a subject can feel two distinct points. Okay? And so if you have some sort of probe with 2 points to it, the smallest distance that you can still detect those 2 points is what's called the two-point discrimination test. So in this last figure, we show a demonstration of the two-point discrimination. Okay. So remember, in our fingertips, we have many receptors that have small receptive fields. That means you should be able to resolve a smaller distance between 2 points. And so here, the two-point discrimination on your fingertips is about 3 millimeters. However, on the forearm, with less receptors, with larger receptive fields, you cannot distinguish with that much resolution 2 different points. So the two-point discrimination test in this part of the skin is 40 millimeters. So for example, if you have receptors with these different receptor fields with fewer, you can only distinguish the 2 points if one is touching each of the receptive fields, like in the forearm. However, in the fingertips, with many small receptive fields, you'll have a very finer discrimination of determining a two-point discrimination. Of course, any smaller than this, then that you will not feel that you will not realize the 2 points anymore. So the two-point discrimination is the minimum distance you can resolve 2 points. And this gives an estimate of the relative receptive fields. And so this concludes a general introduction to these general receptors.